Hello and welcome to another video of grade 11 biology, and the topic that we are going to cover in this video and the next few videos is cell and cell theory. So that's a very important part, and let's start with what is a cell. Now you know that unicellular organisms are there that are composed of only one cell, and then we have multicellular organisms that are composed of more than one cell. So cell can be known as the basic unit of life. Now cells, they can have their own independent existence. That means they can survive alone, and they also perform the essential functions of life within themselves using all the structures that are present inside the cell. So the cell and its components, and what is the cell theory, what's an animal cell, plant cell, the different components of the cell. So that is going to be the focus of this and the next few videos. So before we get started, don't forget to visit our website, that is perfect-scores.com, for more such videos. And if you have any feedback or suggestions, you can mail them at perfectscores89@gmail.com. And this is Preetinder Kaur, and let's get started with what is a cell. Now anything less than a complete structure of a cell is not able to sustain independent living. So we can say that cell is the fundamental structural and functional unit of all living organisms. Now the first person who saw and described a live cell was Anton von Leeuwenhoek. And the person who discovered the nucleus, that is the central core of the cell, was Robert Brown. And lots of other further components were discovered, and that was there because of the invention of the microscope and then the further invention of the electron microscope that could help us to observe really minute things that are present inside the cell. So what exactly is this cell theory? There are a few people that are supposed to be mentioned whenever we talk about cell theory. So these were the three important people. Now in the year 1838, the first person, that is Schleiden, he was a German botanist. He went through and he observed a large number of plants, and he observed that all plants are composed of different kinds of cells. So let me put that down. Plants have different kinds of cells. That was his observation. And then the second person was there, which is Theodore Schwann. He was a British zoologist, and approximately around the same time he studied different kinds of animal cells, and he reported that the cells, they have a thin outer layer, which today it is known as a plasma membrane. So he was the first one to discover and identify that there is a thin layer known as plasma membrane today. And then he also concluded, based on other studies, that in plant cells there is a presence of an extra feature known as a cell wall. So on the basis of this, Schwann gave the idea that the bodies of animals and plants, they are composed of cells and the products of cells. And then came the third person, that was Rudolf Virchow. Now these two people, they had already given a basic idea of the cell theory. But this theory did not explain as to how new cells are formed, and for that the third person was Virchow. Now Rudolf Virchow, he first explained that cells divide, and through division they give rise to new cells from pre-existing cells, which in Latin he called omnis cellula is cellula. So he modified the hypothesis that was given by Schleiden and Schwann, and then the new hypothesis containing points of view of all these three people came to be known as cell theory. So what is cell theory as we know it today? The first point of cell theory is that all living organisms are composed of cells and products of cells. The second important part of the cell theory is that all cells, they arise from pre-existing cells. So these are the two important components of the cell theory. As you can see this diagram, the three people that help in finding the cell theory, Schleiden, Schwann, and then came the third one, that is Virchow. That was about 20 years later, about 17 to 20 years later than what Schleiden and Schwann found. So coming to an overview of what cells are. Now you might have undergone through diagrams or even seen and observed real cells under the microscope. And the common ones are the onion peel or human cheek cells. Now you will observe that the onion cell, since it's a plant cell, it has a cell wall. And this is what we just did, that plant cells, they also have a cell wall, which is an additional feature. The cells of the humans, that means animal cells, they do not have the cell wall. Now inside each cell, there is a structure known as a nucleus that is bound by its own membrane. That is the central part of the cell. This contains all the genetic material, that is the DNA, the deoxyribonucleic acid. Now cells that have a membrane-bound nuclei, where the nucleus is bound by a membrane, those cells are known as eukaryotic. And the cells that do not have a membrane-bound nucleus, they are called prokaryotic. Where there is no membrane-bound nucleus, they are prokaryotic. Now a brief introduction of what this actually means. Pro means initial and eu means new. So prokaryotic, the ones that were formed earlier, and eukaryotic are the more developed ones. Now in both of them, prokaryotic and eukaryotic cells, there is a fluid matrix, a semi-fluid matrix in the cell, and that matrix is known as cytoplasm. So in the cell, you have the cytoplasm, which is the matrix, and the nucleus, and lots of other kinds of organelles are there, or lots of other intercellular structures are present. Now besides the nucleus, the eukaryotic cells, they also have organelles which perform various functions. For example, endoplasmic reticulum, mitochondria, lysosomes, all these. The prokaryotic cells, they do not have any such organelles. And then ribosomes. Ribosomes are one kind of organelles that are found in both cells. These do not have any kind of membrane around them, and we'll be discussing them in the next few videos in detail. So ribosomes are present in both of them. Now within the cells, the ribosomes are found in the cytoplasm, plus in plants it can be found within the chloroplasts, and in the others it can be found in the mitochondria and the endoplasmic reticulum as well, but that we'll do later on as well. And animal cells, they also have one extra organelle called centriole. That is there only in animal cells, not in plant cells. Now cells are of various types, various shapes, sizes, structures, and I'll give you an example of the smallest cell, which is a mycoplasma, whose size is just this much in length, while bacteria can be a little bigger, 3 to 5. The largest isolated single cell is the egg of an ostrich. So that's the largest cell, an ostrich egg. And then you have lots of other multicellular organisms, and within them lots of different kinds of cells. For example, blood cells are there, and we've already covered plant tissues where we did tracheids, vessels, stomata. All these are different kinds of cells. We also did the epithelial cells, then cells of the nervous system, that is the neurons. The nerve cells, you need to know that they are some of the longest cells. And in shape also, they can be disc shaped, they can be spiral, they can be in the form of a cuboid. They may have spaces within them, or they can be tightly packed. So that is all that you need to know about the different kinds of cells that are there. So the next important thing that we need to do is what is a prokaryotic cell. Now I hope you remember what did we do about the prokaryotic cell. It has ribosomes which are there in prokaryotes and eukaryotes both, but it does not have the organelles. Those organelles are absent from a prokaryotic cell. So where are actually these prokaryotic cells found? They are found in bacteria, in blue-green algae, and they're also found in mycoplasma and organisms known as PPLO, that is pleuropneumonia-like organisms. Pleuropneumonia-like organisms. So these are 4 examples of prokaryotic cells. Now these are generally smaller because they do not have so many organelles. They multiply more rapidly. So they are smaller. There is rapid multiplication. And apart from this, they vary greatly in shape and size. The 4 basic shapes that are present in case of bacteria, they are bacillus, which is like a rod, coccus, which is spherical like a ball, vibrio, which is comma shaped, and spirillum, which as the name suggests is spiral in shape. Now the organization of the prokaryotic cell is very similar, while prokaryotes can be different in terms of their shapes and functions. So let's go through this diagram that you can see, and you have a plasma membrane and then there is a cell wall. The inner part is known as a capsule. So this is one organelle that is present, that is plasmid. There is a nucleide also, and it does not have a membrane. So I hope you remember that where the nucleus is bound by membrane, that is a eukaryotic cell, and if the central matter, that is the genetic matter, it is not bound by a membrane, that is a prokaryotic cell. And here you can see it's all jumbled up, there's no membrane. Ribosomes are present. A flagellum is used for movement purposes by a wave-like motion. The central matter is called cytoplasm, and sometimes hair-like structures called pili are also present. So the fluid matrix is there, a nucleus is there, and most of the times, in addition to the genomic DNA, that is a single chromosome or the circular DNA, sometimes there can be smaller circular DNA outside the central DNA. So if this is the central part, let me just use a darker color. So if this is the central part over here, apart from this also DNA can be present outside, which is present in these plasmids. So that is known as plasmid DNA. That means not just in the nucleoid but here as well. Now plasmid DNA is the one that gives unique characteristics to such bacteria. One such example of this unique characteristic is the increased resistance to antibiotics. Now in higher classes, that means after grade 11 or 12, you will learn that this plasmid DNA is used to monitor the bacterial transformation with foreign DNA. And nuclear membrane, it is found in eukaryotes but not in prokaryotes, and only ribosomes are there. There are some kinds of inclusions also, and sometimes a different cell membrane is present which is known as a mesosome, which is only present in prokaryotes. That is a characteristic, and basically it's like an infold of the cell membrane. So these kind of folds called mesosomes are also present in prokaryotes. Now we need to further study about these prokaryotic cells. That means how is their cell and what kind of modifications can the cell envelope get. So most prokaryotic cells, they have a chemically complex cell envelope which is there. It consists of a 3-layered structure. So let me write that down. The outer one is known as glycocalyx, and that is followed by a cell wall, and then comes the innermost, which is the plasma membrane. Now, although each layer it performs a distinct function, together the three of them, they are acting like a protective unit. Now, bacteria can be classified into two groups on the basis of differences in this cell envelope and the manner in which they respond to a Gram test. So, bacteria, they can be identified into two types. One is Gram positive, one is Gram negative. Now, Gram positive are the ones that take up the Gram stain whenever the Gram test is applied on them. Gram negative ones are that do not take up the Gram stain when the Gram test is applied on that. Now, glycocalyx, it also differs in the composition and thickness among different kinds of bacteria. It can be a loose layer known as a slime layer in some, or it can be thick and tough, in which case it is known as the capsule. And the cell wall, which is the middle one, that determines the shape of the cell and what kind of structural support it has. And it also helps and prevents the bacteria from bursting out. Then there is a special structure, that is the plasma membrane, which is the third one. Now, this one is semi-permeable in nature, that is permeable to certain kinds of substances and impermeable to certain other kinds of substances. It's quite similar to the plasma membrane of the eukaryotes. So when we'll do that in detail, you'll be able to recognize this plasma membrane in a more detailed manner. And then I told you that sometimes in bacteria there's a special fold that takes place in the membrane that is known as a mesosome, that is present only in prokaryotes. These are basically extensions of the plasma membrane into the cell. These extensions are in three forms. One is vesicles or tubules, or it can be lamellae. They help in the cell wall formation, replication of DNA, and distribution to the further daughter cells. They also help in respiration, that means exchange of gases, secretion processes, and they also help to increase the surface area of the plasma membrane and the content of the enzymes. Now, bacterial cells, they can be motile or non-motile depending on their locomotion abilities. So motile means the ones that are capable of movement on their own, and non-motile the ones that are not capable of movement. If motile, they usually have a thin filamentous extension from the cell wall known as flagella that we just did in the diagram. Now, bacteria, they show a lot of range and variety in the kinds of flagella that are found in the numbers of flagella. Now, this flagella, it's composed of three parts. One is the filament. The other one is the hook. Now, filament is the long hair-like structure that you see. Hook is joining the filament and the basal body. Now, the filament is the longest portion from the cell surface to the outside. Now, besides these flagella, there are few more other kinds of growths like pili and fimbriae. Now, these are also present. These are surface structures, but they are not used only for locomotion. The fimbriae are basically small hair-like or bristle-like structures, and sometimes they help to attach the bacteria to any kind of surface. So basically their function is attachment, not only locomotion. So that was a brief description of the cell envelope which is there in the prokaryotic cells. And now it's time to learn a little bit more about the ribosomes that are present in prokaryotic cells and the inclusion bodies. So in prokaryotes, ribosomes are there in the plasma membrane. This is where they're found. And they're about 15 into 20 nanometer in size. And they're made up of two units. One is known as 50S. One is known as 30S. And they both combine and form together a 70S unit. So that is just the terminology that is used. The ribosomes are used for synthesis of proteins. That is their function. And several ribosomes, they can attach to a single messenger RNA. Single messenger RNA, and that can form a polyribosome or something that we can simply call a polysome. Now the ribosomes of the polysome, they transfer this messenger RNA into proteins. That is how these proteins are made. And then comes the second part, which is inclusion bodies. Now this is just a reserve material that is stored. The excess material storage is done in these inclusion bodies. They do not have any membrane, they are just free lying in the cytoplasm. Some examples are phosphate granules, cyanophycine granules, and glycogen granules. And then sometimes gas vacuoles are also found. So these are some examples of the reserve materials that are stored in the form of inclusion bodies. Now the next thing you need to study is what is a eukaryotic cell. So that is the structure of a eukaryotic cell, and lots of different structures you can see apart from ribosomes. The eukaryotes include all protists, plants, animals, and fungi. All these four groups, they are part of the eukaryotic division. In eukaryotic cells, there's a lot of compartmentalization of cytoplasm because there is presence of membrane-bound organelles. That is the most important characteristic of a eukaryotic cell. They have an organized nucleus that has a membrane around it. That is also one important part. And lots of different eukaryotic cells, they have different kinds of structures. For some are used to transport materials from place to place. Some are used in locomotion. For example, let's say bone cells or muscle cells, they're helping in movement and locomotion. Some, they perform cytoskeletal structures. So lots of different functions are there, and most of their genetic material is present in the nucleus in the form of structures known as chromosomes that we'll be covering in detail in the next few videos. Now all eukaryotic cells, they're not identical. Plant and animal cells are totally different, and I hope you remember the difference between plant cells and the animal cells was the presence and the absence of a cell wall. Plant cells do not have a cell wall. It is present, plant cells, they have a cell wall. It is absent in animal cells. So animal cells do not have any extra cell wall. On the other hand, in animal cells, there is an extra thing known as centriole, which is not present in the plant cells. So the plant cells, they do not have any kind of centrioles in them. So two important differences between plant and animal cells. So over here in this diagram, you can see example of a eukaryotic cell, and within this we are just going to study briefly plant and animal cells. So remember these two differences that we've just done, the presence and absence of cell wall and centrioles. So this is an example of a plant cell. And one more thing you can see is there's a huge vacuole that you can see in the central part. This one, that's a huge vacuole. And in the plant cell, there is also a cell wall that you can see over here. And then there's an adjacent cell wall. There is no centriole to be found. It has the cytoplasm, it has the chloroplast, ribosomes, mitochondrion, the plasma membrane is there like in all cells, the nucleus, Golgi apparatus, and chloroplasts are also there in plant cells. Now the function of chloroplast, as the name suggests, they have chlorophyll and they help in photosynthesis. And there is smooth endoplasmic reticulum, rough endoplasmic reticulum, and lysosomes as well. Now the different one is the animal cell that you can see over here. It has a plasma membrane, it has a centriole, and all the other bodies are there: cytoplasm, nucleus, nuclear envelope. It has the endoplasmic reticulum, Golgi apparatus, but it does not have a cell wall. And it has a centriole. So in the plant cell, centriole is not there, cell wall is there. In animal cells, cell wall is not there, centriole is there. So that is all that we'll be covering in this video. This was just a basic introduction to plant and animal cells. And we've covered what a prokaryotic cell is and what are the different components. Now in the next videos, we'll be discussing about the components of a eukaryotic cell. Different components starting from cell membrane. So thank you so much for watching this video. I hope it was useful and you have a good idea of what cells are, what is cell theory, and what are the different kinds of cells. Don't forget to visit the page perfect-scores.com for more such videos before and after this topic, and any suggestions or feedback you can mail at perfectscores89@gmail.com. So thank you so much for watching this video.